annotations, then multiple alignments, sequences, then it can also be used to build a phylogenetic tree, NGS assemblies, and the likes. So Eugene, as a tool, incorporates numerous biological techniques that is used in bioinformatics. And this tool has some pre-built, sorry, let me just change this to a slideshow. All right. So, like I was saying, hello, can everyone hear me, please?
Yes, we can hear you.
Yes, we can hear you.
We're following, ma.
Okay. So Eugene has some pre-built tools which allows it to carry out all these functions, bioinformatic functions, and in addition to sequence alignment and other things, it has a graphical user interface, which enables people who are not so savvy in bioinformatic techniques. It makes it easy, like it's an automated software that can be used by beginners to understand the bioinformatic techniques. So Eugene has a tool is in form of an application of software, like I mentioned. Then it can function on different OS, Windows, Mac, and Linux, and it is a free software that is available online through this link. The key features of Eugene is that it can be used to view, edit, and annotate the DNA, RNA, and protein synthesis. Also, it can also be used to edit and align multiple sequences. Because of the visual interface, it can also be used to view, like after aligning the sequences, it has a sort of a kind of interface that it's easier to detect differences in alignment. Like when you align your sequences with Eugene, with the kind of the visual interface it has, it's kind of easier for you to detect reserved region on sequences. And also, in addition to that, it can be used to detect or build 3D structures. And also, it also has one very, very important feature, which is a workflow design. These aspects too, I haven't really explored it much, but I know workflow design is like you having your Eugene carrying you through your bioinformatic technique right from the beginning. After putting in your raw data, then you align it. Then you can decide to draw your tree or carry out other bioinformatic techniques. Then you have this saved. Like it's something you can save and continue from where you start, like a kind of workflow. So Eugene has this feature. Then, like I mentioned earlier, Eugene can be used to create protein sequences. Like you are having your sequences somewhere and you want to like type it in. You're not you're not uploading a file. You like just have a type sequence that you want to see what can happen. You want to play around the sequences. You can use Eugene for it. It has a feature for it. Then even within your sequences, you have a particular sequence that you're interested in that you already have available with you. You want to rapidly search within your bulk of the sequences that are aligned. You want to see where the exact position of a particular short read. Then you can easily put it in, and Eugene will bring it out for you without going through much stress. Then, like I earlier mentioned, it can be used to align multiple sequences. Then, since you can easily, with the visual interface, you can easily use it for variant calling. By the time you compare your sequence with a reference genome, then you can know the conserved region and be able to know where, like variant calling, like Eugene can be used to how do I say it, to detect differences or changes or what well, like to be able to get the main conserved region in your sequence, which is mostly the reason why you're sequencing for in the first instance. So also, it can also be used to, without having to leave Eugene, you can search through online databases directly when you are on Eugene, the protein database and other databases. So it can also be used to share, to create and share your stored data, like a lab database can be shared within Eugene. Then detection of your open reading frame. Then plasmid can be constructed and annotated using Eugene. Also, cloning, in silico cloning can be designed using Eugene. Then these are the sources. So basically, that's the work Eugene does. So I would just like to open something so that you can see it. I like not really hands-on, just to see the Eugene interface itself. Just give me a few seconds, please. Okay.
Okay.
So this is a sequence. This is just a view of a sequence on Eugene. And this is sorry. This is the object view. And here, as you can see, you have different annotation telling you where you have your ORFs from here. These are your sequences. And within these also, if, for instance, now, if you intend to like, let me just see. Okay, now let's say I'm looking for, like I want to look for a particular sequence, like the location of a particular sequence. I can easily click, there are information here where that you can work with. Here you can type in your sequence pattern. Let's say for, like you decide to, you're searching, if it's a much bigger sequence, you can search for a particular read on your sequence. So you can see that it's bringing it out, like you can see the location of that sequence. So even if you have a long read and you have a sequence you want to search within a long read, you can easily type it in here and it will bring it out directly. You see the location of that particular sequence. That's one of the functions. Also, for the alignments, let me open another file for the alignment.
Please, sorry. I just want to know, is this the Eugene platform website?
Yes, yeah, this is the Eugene platform. I think I said that before I started.
Okay.
So this is the Eugene platform. Should I go over it again? You have to download
Please, can it be
it.
downloaded?
Yes, sure, it's available. It's online. It's free.
Okay, thank you, ma.
So when you download it, but you have to have all these information on your system. Like you can go to NCBI and download different sequences like you were taught in our previous sessions. So when you have your FASTA file that has multiple sequences that you want to align, once you have it saved on your system, so all you have to do is just to come to your Eugene, search for the file, using the file location. So I know this particular file I'm working on that I just opened already is here. So that's the COI and that's the alignment file. It has multiple sequence in it. So once you open it on Eugene, you don't have to do any work to say like to align. It's automatically aligns it. It brings the alignment view for you. And yeah, that's because I've actually worked on this before. Let me see if I can refresh this so that you see it. But once it's not like how you do anything. Once you have a multiple sequence in a file, like your faster file that has your raw data of different sequences. If you're having just one like a single sequence in your file. That was the initial one I showed you. That's the view you're going to have. It's going to bring it in front of the line. But once you're having a multiple sequence, you open it on your Eugene, it will automatically generate an interface that has aligned the sequence for you. And as you can see, if the alignment is very, very easy to navigate through. You can see with the nitrogenous bases are colored in different ways, and up here you can see on each column there's a graph to tell you the percentage content of the nitrogenous base in each column, and with the color differences you can easily detect where you're having changes, where the sequences differ. So also, when you have your sequence like this, all you have to do is just to click on your phylogenetic. There's an option there like a tree-like structure. You just click on. Once you click on it, and you can choose the type of tree you want. This is the common one anyway. So there are other options there, and this is the one I'm more familiar with. So you just okay.
Please, ma, I have a question to ask you.
All right. That's right.
Okay, ma, my question
Sorry,
is this.
please.
No,
Sorry,
ma.
please. Sorry, please. The presenter, Mrs. Suliat, please tell Mr. Daniel to off his mic. He's interfering with your presentation. We can hardly hear what you are saying.
Oh.
Yes, Mr. Daniel should please off his mic.
Please, Mr. Daniel, could you please help us mute yourself? Has it done so already?
I think he left
Yes,
already.
thank you.
Okay.
On the contrary, you can mute everyone, please.
Oh, then the moderator will do that for us. Okay. So this file I opened has multiple sequences in them. So once you open a multiple sequence using a Eugene, like from your Eugene app, you open the file, you select the file containing your FASTA file containing your multiple sequence. It brings out this visual interface showing your sequences already aligned. And as you can see, these are the number of sequences that we have here, and this is the alignment view. So you can easily also build your phylogenetic tree using this platform. You just click on the tree section and you have different options, but I'm more familiar with this, the neighbor joining, the free neighbor joining. Then you build your tree and you can see. And also your tree can be edited. This is your phylogenetic tree. So this can also be edited. Once you click on it, you can give different colors to it. You can edit it as you want to suit your view. So you can see once you click on the text, you're already clicking on where you want the color change to be done. So you can click on the color, then you see different options. You can decide to use red. So there are so many things you can do here. That's just for the alignment. Also, you can view your SAM file. Let me just open another file here. Okay, I'm unable to open this particular file. So let me just open another file on the 3D structure, and there's so many things you can actually do with your Eugene. And I'm still exploring. I'm here to exhaust all the functions of Eugene, but it's a very, very easy to use tool for beginners on bioinformatics. It doesn't really need any special um, knowledge of the by the all this technology, all these IT skills and like the likes. It's just a direct tool. Once you have your file loaded on your system, all you just need to know is to know the file, like the kind of file. If you're working on alignment, you should know that you want to align your sequence. You should know you have to have your file, your faster file, having the alignment, all the sequences you want to align. So once you open on Eugene, it will bring it out. It will bring the aligned sequences out for you. Okay. So I think that is basically those are the things you can do with your Eugene. You can also you once it's over time as you use it, then you get to see more other features it has. So at this point, I think I can stop. If there's any question, then I would be glad to answer.
All right, Mas. I have a question.
Okay.
Please, ma,
I have a
can
question.
I have
I
a question too.
how
Okay.
can I from Google access this Eugene? Because it's like you're opening already
Okay.
stored
I
file.
shared let me open the
I have
I
already
shared
shared
the
the link
link.
to download the Eugene on the chat box. You can check it there.
So this is the link.
It's
So
already
it's
been
the same
shared.
as okay.
I have a
All right.
hello.
Yes, I'm with you, sir.
Can this region be used for primer design?
I'm not sure. I am not sure. I don't think it's one of. I'm not sure. I don't think so. I don't think so. No. Well, you could also find out, but I don't. For now, from my own knowledge, the little knowledge I have of it, I don't think it can. I am not aware it can be used for that.
Hello, ma'am.
Okay,
Yeah, please, can we get the slide for this lecture?
definitely. It's going to be made
Thank
available.
you.
You're welcome. So, Mr. Asher, I think I'm done. I don't know if you still have
Yeah,
questions.
thank you so much, Ma.
This morning, I want to
I
ask a
also have a
question,
question
please.
actually. So, but
Okay.
before that, I would like to Thank you so much for the introductory part of Eugene. To be frank, I think I think on Monday when we were having the first session, a question was asked regarding some tools that can be used for phylogenetic tree. So and for alignment purposes too. So, and I think from your presentation about Eugene, so it is now obvious that one can use Eugene without having to trim, having to go through the stress of waiting, realigning, doing this, doing that. Another advantage for Eugene is that you can use Eugene as a user of micro Windows, Mac or Linux. So you can use Eugene as a tool for your bioinformatics analysis. So this is personally I have not tried with it. From what has been presented so far, I think this would be my newfound love. So there is no time to stress myself on trying to trim and other stuff because I have everything on a platter of gold on a very one single point. I can do the phylogenetic tree, and during the phylogenetic tree that was shown to us, the option of neighbor joining tree, there is another option that I'm also using for my full genome mitochondrial study, which is the IQ tree. So every researcher has reasons for using these different tools, different parameters, or different analysis. Even in the neighbor joining tree, it will ask you: is it chimera two parameter? Is it Jukes and Cantor? Is this or that? So if you do not know what you are actually working or looking for, you might have a problem doing that. So maybe subsequently, if time permits, we would have a full course on phylogeny, which is one of the things that you use multiple sequence alignment for. So now another thing that was mentioned during the presentation is the variant calling. I'm actually doing a work presently that requires me to do single nucleotide polymorphism. So with Dr. Aisha Awaji taught us about multiple single nucleotide polymorphism. She hinted about it on the first day. So, and I saw that this tool Eugene is another tool that can be used conveniently. That's the word. Conveniently, easily accessible, and very straightforward. So you can use it to do a lot of variety of work. So thank you so much for taking us through this analysis. Now also so that I won't jump before presenter. So I noticed that she mentioned about SAMP or BAM files. So these are file titles. There are different file formats that you'll be coming across in bioinformatics analysis. So the commonest and the very cheap one, or let me say, is it cheap? No, is the commonest one is the fast A file or the TXT file. So when you are dealing with next generation sequencing results, you'll be talking about BAM files, SAM files, VCF files, depending on the type of analysis you want to perform. So this Eugene comes in handy. So which would make me has made me even fall in love with the software itself. So I would rather say that, just like I said from the beginning of the class, that we are here to benefit from each other. I've learned so much just from these few minutes of presentation. So I think my question has been answered. So thank you so much. If we have any question before we move on to the next session, so we can quickly have it. You can either type it in the chat box, or will you unmute yourself to ask.
Yes, I have a question, please. Good morning.
Good morning.
Yeah, I'm very, very new to bioinformatics. So I'm wondering because I've come across this FASTA file. I couldn't open it on my laptop. So I want to
Oh, which
know
file? Which
the kind
file?
of program FASTA, FASTA.
Oh, okay,
You
okay,
know,
okay.
fast. I want to know the kind of program I can use in opening it, or do I need a different software to use to open that kind of file?
You can, are you with the file right now?
It's on my laptop, but I'm speaking with you on my tablet, so I can access it right now.
Um, just the next time you get to the system, you can simply just right click on the file and view open with notepad.
Okay.
Just right
Okay.
click and open with notepad. Every computer system has notepad, so you'll be able to
Yes.
view
Yes.
the file through notepad, so it is easily viewable. You don't need any software to pass a file.
All right. Thank you so much.
You're welcome.
Please,
Yo, ma,
I have a
please. I
question.
have another
I have a
question.
question.
I
I have a
also have a
Please, the
question,
moderator,
please,
please go to the
if you
message
permit me.
section of this Google Meet. Please, can you go to the message section of the Google Meet and see there are questions there, please?
Ah, okay, okay,
And
okay,
I think it's of benefit to every one of us. Thank you.
okay.
I have a contribution to make.
Also,
Please, I
sorry,
have,
if I may chip in, please, we might raise our electronic hands so as not to make the questions crowded. Everybody will not be talking at once.
Okay, 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 okay. You can raise the hands and then I would call on the question. So, but let the previous Mrs. Is it Miss Aja? I'm sorry, I didn't get the name. You can finish your question so that we can.
Yes. The order of my question is from the lecture she just gave that Eugene is you can use it for so many bioinformatics analysis. I know that bioinformatics basically is all about DNA and sequencing and all that. So I'm wondering, how do you do this sequence? Because she spoke about if you have so many sequences, how do you get a sequence? How do you align and all that? Like if I'm working with because I'm into plant sciences, if I want to get a sequence from a plant, okay, let's say I'm looking at mutation in a plant, and I want to know what makes this plant be able to, withstand this kind of mutation. How do I generate a sequence from from it? Like I'm confused about what to do to extract something that you say is a sequence and how you go about analyzing it.
Okay, doctor.
Okay, sir.
Okay. Good afternoon. Okay, let's be answering one after the other. Okay. Okay, let me quickly answer that. I think doctor is on. So
I
this.
didn't get the question
Okay,
right.
okay.
She wants to know how to get, like, how we get data for Eugene. Is that the question?
Yes, yes.
Okay. Okay, usually um, data are gotten from sequencing anyway. So, and um, usually sequencing is carried out. I don't know her background anyway. Sequencing is done to know the nucleotide content of a particular, like the nucleotide content of a DNA. So once this is done through, it depends. We've actually probably would have had our next generation sequencing presentation before this. That would have helped a lot. So when you have your sequence from your sequence result due from your sequencing, which ever of the sequencing technology decide to use, these data are called your raw data, and most times they are stored in form of your faster format. Like these data are saved, and you can easily access data even without having to go through sequencing. If you want to carry out research, there are databases online, like your NCBI is a data source, and other data banks too are there. So you can easily obtain sequence of interest from this online platform, and you work on them. Like you want to compare, like for instance, now saying you want to compare ATPase, that is like adenosine triphosphate, is an enzyme. So you want to compare ATPase of different organisms. So all you have to do is to go to your NCBI, download ATPase, and even for ATPase, there are different type of ATPase. So you have to be specific. You have to be specific. Like prior to doing that, you have the knowledge of what you want to do. So there is hardly any sequence that you not find online there. So you just go there, you type in your sequence of interest. So now that your interest is to compare, for instance, to compare the differences in the ATPase of different organisms. So you know you have the organism of interest in mind. So all you have to do is to download them. So by the time you download, you select. That's we've been taught in our previous sessions. I think that's the first class. So you download those sequences instead of now using bioedit like you we were taught in the first session. So you can just download those sequences as your FASTA file. Then once you have it on your system, by the time your UGene, by the time you open your UGene, which is has been installed on your system, I'm assuming, so you can just go to the file format of your UGene, then click on that particular FASTA file that you've downloaded, which contains different sequences from that you've downloaded, your sequence of interest. So by the time you open your UGene, it will bring out, it would have already aligned those sequences for you. And easily with the visual interface that we have in UGene, you can easily detect the differences. Like you must have an interest, you have an objective for comparing them anyway. So probably you want to know maybe the way this is functioning, have some function, why some ATPase could function, could have some function, why some others don't have. Then you'll be able to easily see the region of differences and you probe it to see, okay, is this what is behind the notable differences that functions that you've seen in there as you've seen earlier, which made you downloaded the sequences for comparison. So I think that's just my own simple way of answering your question. Mr. Ashraf can do better.
Okay,
Yeah,
could I
please, I have question.
No.
I have a
Please
question.
raise up your
Hello.
hand if you want to ask questions or type it out. I have identified
I,
some of the questions so that it won't be
I
rushed.
didn't see the
Please,
option here. I didn't see the
you
option
can,
here to raise hand.
or you
Okay,
can type it out.
I want to add to the question Miss Obi asked.
Okay.
Okay, from her question, I think I understand she's still very new to bioinformatics, and she's trying to understand how does she obtain sequence. Maybe as a researcher, she wants to obtain the sequence to maybe a plant or something she's working on. Well, Miss Obi, if you want to obtain sequence, it's bioinformatics starts from the
Laboratory.
DNA preparation stage. So to obtain a sequence from whatever you're working on, whether it's a plant or an animal, you have to do your DNA prepping, which is where you collect your DNA. Then you have to, if you don't have a sequencer, you can send it to companies, maybe genomic companies that have sequencers. They now use the DNA that you've sent to them to sequence the organism. So if they by the time when they're done with the sequencing, they send you the sequencing data, which you could now impute into maybe the and maybe and whatever platform you want to use, maybe the NCBI or maybe Eugene or whatever, or maybe the protein data banks. So when you impute your obtained DNA sequence, which you've gotten from your plant or whatever, you want to compare it to what's already existing. So you now impute it and do your blasting, and maybe you want to do sequence alignment. So that's just it. So you have to start with DNA prepping to obtain your DNA sequence for your own particular organism. So you cannot use it to compare to whatever they have around the world. So that's basically what I understand from your question.
Thank you so much. That's concise
Can I
and
add something?
as comprehensive as it should be. Thank you so much. Um, we have some questions listed here already. So one of the questions is about can we? They needed a walkthrough of the process for the Eugene. So our resource person has already taken us through the process. So she has downloaded the sequences already, and then she just uploaded it on Eugene, and then the results are there to view. So I think it's that easy. So the second one is, are we going to still going through the are we still going through the file, that is the API file format, FASTA format, FASTA format, and the likes? So are we still going to be taking through the sequence files, when to use them? Well, I think
Yes.
we are
Yes.
still going to talk about. I think the resource person is going to take us through in the next session, which is the NGS session. So don't worry, we'll still be taking you through that. Another question is Eugene for sequence alignment instead. Yes, of course, she showed us how it was done with alignment, sequence alignment, and then can we use it to this unigenes? I don't know that one, so maybe it can be asked. So if we have more questions, we can pen uh, the
If I have
questions.
question,
Okay, okay. I think we can take five more questions so that we can move on to the next
yes,
session.
please, I have question. My
Okay, go
question is
ahead.
this: I want the
I have a
I want
question.
the okay. I want the presenter to take us to Google because I've opened the Eugene environment. It's not what she showed us. Let her go to Google, show us gradually how to, do this thing because I've opened the environment. It's different from what the presenter showed us. Please, I want her to go back, take us to Google, even if it is from the link. Show us how to do it gradually because she was fast and we are not following.
Okay, did you download it? Let me ask. Did you download the software? You have to
No,
download
I
it
didn't
and
download.
install it.
No,
You
yeah,
download
yes,
and install on your system to see what I showed you. If you don't do that, you can't see
ah,
that interface yet.
ah,
Yes.
I thought it's online. When you click, you can
No, no,
access.
you download it and install it on your system to be able to see it that way.
Oh, oh, okay. Let me do that. Sorry, I'm trying to fill up. Yes, yes. It's practical. I'm sorry about that. I know. I'll try to observe.
Okay. If you follow the procedures that has been described, it's very easy. It's not hard once you have it downloaded on your system.
Okay, I get it
Uh,
now.
good morning, everyone.
Good morning,
Can I
sir.
ask a question, please?
Oh, please go ahead.
Yes, please.
Okay, mine is more like a contribution because, okay, personally, I have peripheral understanding about molecular biology, bioinformatics, and all of that. So, the whole essence, what is actually the whole essence of doing sequence alignment, the phylogenetic tree? Maybe I have an interest. I want to study a particular DNA sequence, and then I am doing sequence alignment. After getting the sequences that align closely to my own test sequence, what next? Like what next? What am I expected to do next? So I don't know if we can get A very good question. um, I don't know if we can get maybe a research work that has been done and published, because I heard you say you are doing some work or you have done some research. So can you send a link? Can you share some of your work, some of the work that you have done, so that we'll actually have a clear understanding of why we are learning bioinformatics? Because yes, I know in the course of learning bioinformatics, I get to learn sequence alignment. I know what it means for this sequence to be closely aligned to my test sequence. So what to use? What am I going to do with the knowledge of this um, alignment? Having gotten the sequences that align perfectly or closely to my own test sequence, what next? So I don't know if you can probably share a research that you have done, a
Yes.
published work,
Thank
so
you.
that we can, yes, so that
I will
it will
share a link
actually direct us on the
to
essence of
my
learning.
Google Scholar page. You can have a feel of some of the work that has been done. So, in particular,
Okay.
the first one, I would have loved to share my screen, but I wouldn't want to distract our presenter. So, for example, the first one on the list, genetic relationship of four armyworm population that invaded Africa and Asia, genetic diversity of Bemisia tabaci in Nigeria, uh,
I
complete
can't see what you
physiology.
are showing. I can't see what you are showing.
I'm not sharing my screen just yet.
Okay, let me just stop presenting so that you can do that
Okay, okay, okay.
if it's possible.
So
I
actually,
think you
it's
can.
a very, very good question that was asked. So, uh, and we ought to have had that introduction right from the beginning. So
Will it be possible for you to run down how you extracted this DNA on your own because you have worked on it, and then the information you gathered from your research, just a summary of it?
I didn't get you.
I said whether it will be possible for you to summarize how you extracted this DNA from
Oh,
the source, and
that,
and then what information do you derive from your work, just a summary of it all.
okay. Well, I don't think it's
Agree with what you just
theoretically
said now.
possible to take you through the DNA extraction process, but can you see my screen?
No, no.
Not yet, not yet.
Okay. How about now? Yes.
Yeah,
Yes.
yeah.
So, one of the ways to sequence your DNA is first of all get your sample. Maybe it is blood, maybe it is tissue, or maybe it's anything you want to sequence. Maybe it is plant, maybe it is the root or the leaf or the stem. Anything you want that is biological that you want to sequence. So you take the sample and then you follow your lab protocol to do the DNA extractions. So you get the DNA sample and then you send it to the sequencing company. I know in Nigeria there is Incoba, there is African Biosciences, there is 54 Gene, there is there are many. I think like 4 or 5 in Nigeria that I know of. The sequence that they do DNA sequencing. So, uh, is my screen still visible?
Oh, no, no,
No,
not
it's
again.
not.
Or
Not
I
at
Please one
all.
question.
let me try sharing again my entire screen.
Can I ask one question based on what you're saying now?
Okay, they said you can do it in Nigerian Institute of Medical Research also. I don't know what is happening here. Why I can't share my screen just yet. But please hold on. So you get all the sequences sent to the sequencing company. So once that is done, and then you get your ACGTs in the form of your sequences, and then you proceed to do the analysis. So the analysis is whereby you get the information from your sequences that you need to work on. So that is where the work starts from. So now this is when you now be using the bioinformatic analysis skill. So for example, my first paper on bioinformatic analysis is on Bernicia Tabashi in Nigeria. So let me open, hopefully I will be able to see it here, but I doubt it, it's paid journal. Let me check this one.
That's so good. Have you finished the film? No. It's okay.
Please
Take it.
mute yourself so that we can hear the comments.
I think I have it downloaded, but it's not that near. Okay, the access is through my institution. So, but uh, I'm not in the lab now. Okay, what we will do is that I will send this file when I get back to the lab today. I'll send this file. I'll download it. I'll send it to the group for us to view. So actually, we had this also similar situation in the last session that I had to explain through the process of publishing this journal. So this second one, let me look for one that is free. Okay, I think this one. Yeah, this is the one of my latest. Okay, so we were able to use this what we are teaching you now: alignment, sequence annotation, and mapping for the analysis. So the one on mosquito, South Korea mosquito. See one. Okay, sorry, it says page 2. Okay, yeah, I've seen it. Hopefully it's okay. I'm so sorry, my network is very a little bit slow. So this type of the analysis you do. So number one, we were able to do the phylogenetic tree of this from these samples. For example, in my work on the South Nigerian Bermudian tabasi whiteflies, I collected a lot of samples from Nigeria, and I checked their relationship with one another. Oh my god! Unfortunately, anyways, maybe before the session begins tomorrow or on Friday, I'm going to explain or take us through the process of doing the analysis. So as to save us the time and the stress. So I'll do that when I get back to the lab and uh, send us the files whereby we can see the wonders that can be done with what is being taught to in the class today. So, for example, this is a work that was done in
Finish.
2020. So we also use what we've been able to learn today in class to do some of the analysis. One is the phylogenetic tree, and also we did some work on the population genetic studies using the nucleus TPI gene. So we worked on fall armyworm, which is the Spodoptera frugipada. For those who are in plant related entomology, or if you are in your plant science, so you would have known about Spodoptera frugipada. So these and other publications that we use this bioinformatics analysis tools to do, I'm going to send it to us in uh, chat box in. So, for example, here we use the full mitochondrial genome of giant water bug Lethoceros delorli. So we use this phylogenetic tree, how to draw phylogenetic tree. We use it in doing that. So that was why I was hinting on the fact that I didn't know that Eugene also used IQ tree. If you check this particular section, hopefully I can hold it. Uh, but I didn't expect I'll be doing this presentation today actually. So but notwithstanding. So this we use IQ tree using this model for for the analysis, and we're able to get the results. But notwithstanding, don't let me waste our time. So I'm going to send all this file to us probably in the next few minutes. I'll head down to the lab and send it to us, and then we can have a feel of what it means to have these particular skills. So thank you so much. I think we can have a little bit of
Hello, hello, Dr. Ashraf.
yes, sir.
Dr. Ashraf. Hello. Please just can you just spare me just a few seconds? Some of us are listening from various part of the world, like me. I'm listening from Saudi Arabia now,
Okay.
and we are all beginners in this aspect of. If you want, maybe you can just help us to simplify this aspect. You can see that some people are still missing some things. Even some of us that have not even talked, we have missed a lot. We are beginners. We are just like just struggling to catch up. Maybe you can just try a video, basic aspects of it from DNA extraction. You know our problem now as beginners. Maybe you can just simplify and make a YouTube and just post it for us so that we'll be able to be carried along. Thank you.
Okay. Okay, sir.
Then, in addition, I think Friday you are mentioning something like Thursday or Friday. Particularly, maybe I will not be available on Friday. If that will be possible on Thursday, I will appreciate.
Okay. Okay. The
I
timing.
have, I have a question.
Okay. Any other question?
Yes, I have a question.
Please go ahead.
I have a question. Actually, even though I know that DNA extraction is it's not like something that someone can do a video of. My question is, assuming assuming someone wants to do a study on maybe research on cancer or maybe or disease causing organisms.
Is that Ekram? You can please mute yourself. You're interfering with the class.
Okay, go
Assuming
ahead.
what
Yourself, you're
someone wants
interfering with the class, please.
assuming what someone wants to find a cure or something, how something mutates, or if a particular organism can be able to cure a particular something in, maybe there is a disease or something, how does that person use bioinformatics for it? Like, what does the person do? How does it start? What is the relevance? As in, how can it help? Like, what is the relationship? How can you use bioinformatics to uh, do cancer research? To do what's it called?
Now
To
I
to
understand
search for,
where you are heading to. Well, there is
yes. Do you just, is there a target? Is there already an existing sequence, a target sequence that you would now, you will know this is what you are looking for? Is it like it's somewhere there is a bank, like a bank that has sequence for a lot of things, that when you are now you will now, you say this is the target of the nucleotide you are looking for?
Exactly.
Thank you so much for the questions. Let me take it from the previous comments. Thank you so much, sir, for the input. Regarding the gradual process of DNA extraction. So, because DNA, there is two different aspects of bioinformatics. We have the dry lab and the wet lab. So, right now, from the first session, our presenter talked about the explosion of data. Before, it used to be there is no enough data. Now we are having over explosion of data. There are lots of data that are not yet explored to give answers to human problems or human diseases. So that is why we need a lot of researchers in this aspect of bioinformatics. This is just like I said in the beginning. This is just the lowest of the lowest beginning that we can start from. But the good news is you can move from level 0 to level 100 in a very short time once you are committed to do the work. We have shared the link on our Telegram page on our previous sessions on the genomics. We call it genomics 1. So whereby we took us through the rudiments, the ways we can do alignments and the likes. So these sessions are just a continuation of the first sessions. I will provide the link again. We will provide the link again and send it to us on the group chat so we can go through that. The DNA extraction protocol. There are different protocols to DNA extractions. Like now, I've been in two different labs. I was in another lab during my masters, and I'm in another lab during my PhD now. Just in the same institution, we have different DNA extraction protocols. So if you are working in a wet lab, which is whereby you'll be dealing with extracting the DNAs, extracting the RNAs, doing western blot, southern blot, or any blot type, doing molecular biology analysis, sequencing, that is the wet lab. The dry lab is what we are doing here. The lab analysis, whereby you're going to analyze the sequences, infer information from them, pick out the solutions from from problems that might come up from your plant or anything. That is what we are doing in this class. We are the dry lab section, so it's not really the wet lab section. So I think that answers that regarding the process of DNA extraction. What we are doing here is to analyze the DNA, the ACGTs that we get from our DNA extraction. We want to infer information from it, so not just the letters that we see on the screen. So then, moving on to the next one is there a template whereby you use bioinformatics to solve a particular ailment or a particular cancer or the likes? So we are moving towards that era now. That is the advanced stage, which is the peak of what we can do. For example, even at this stage we can also do so to some certain extent. In our recent publication on Spodoptera frugipada, which I said I'm going to share with us, so we talked about some genetic variation. This genetic variation between Spodoptera frugipada from Africa or from the ones in Asia. So it tells us once we sequence their DNA and we study the DNA, we can notice the differences between the two DNAs. So we can see that, oh, maybe there is a mutation in this particular gene in the DNA of the ones in Africa that is different from the ones in Asia. So once you are able to get that information, we can approach controlling that particular pest, which is a pest. We can control it effectively. For example, I presently work on mosquitoes. We've noticed that some mosquitoes are resistant to a certain group of insecticides. pivot rods. So we've noticed that in their gene, we've noticed a similar pattern that was not there before. If you sequence these mosquitoes from 10 years ago and you sequence the ones right now, you will notice a different pattern in their DNA content. So there is a particular search section in their DNA that is different from the ones that are not resistant to this particular insecticide. So that is an area of mutation that will be studied, that will be known. So this is where it will be giving us information of what is really happening. Like for example, now we all know that the coronavirus of 2019, they said it is rapidly evolving, rapidly evolving. Why is it rapidly evolving? Because the coronavirus itself is trying to evade the control mechanism that the human beings are putting across to it. So that is why they are trying to evolve. So if we do not have that knowledge of bioinformatics, the vaccine that was later discovered that was later developed from bioinformatics skills also and other epidemiology skills would not be applicable to control or serve as a good vaccine candidate for coronavirus. So that is the essence, that is the importance of what we are really doing in this bioinformatics that we are trying to begin the process right from now. So due to the vastness of this biological data, it is very vast, very big. We know that the human gene as the human genome is about 3 billion, 3 and 8 zeros, 3 billion nucleotide sequences long, and they discovered that there are about 20,000 genes in the human body. So imagine that volume. So now being able to infer or take information from that particular section is what we are trying to do in bioinformatics. Another example, we all know this popular actress Angelina Jolie. So she was able to detect she had a cancer gene inside her. She passed through three cancer, or what do they call it, radiation sections before she went through meta, is it meta, what's it called, I've forgotten the name, that she went through this surgery to remove the faulty genes from her body. So that is one of the things that bioinformatics is trying to do, personalized medicine, whereby it is not everybody that artemether and lumefantrine will work for to cure malaria. Or so once you are able to discover the gene, for example, our first presenter in the first session talked about this also. The UK is now sequencing the full genome of babies. So once they've been able to have that information about the genome of babies, anytime there is a disease occurrence in that baby, they can be able to trace it directly to where the problem is, and then they can be able to address that problem. So this is one of the benefits among a lot of numerous benefits that we are going to benefit from bioinformatics. I know as a beginner you'll be wondering that where are we going? We are just doing alignment. What's the essence of the alignment? This is the foundation. If you do not have the foundation right from now, you might end up having a problem when you are dealing with other complex situation in the very near future. So that's why we are making the basis for us in order for us to stand on the shoulders of giants and then to do wonders with bioinformatic tools. So thank you so much for that question. I hope I've been able to educate and not confuse you more on what bioinformatics is.
One more, one more question. Now I understand.
Thank you very much,
Welcome.
Akintola.
I understand
Thank
a
you
bit.
very much.
So I think
Thank you very much.
now we can move on
I have
to
one more
we'll
question.
pause on further questions and comments for now so that we can complete what we embark for
Please,
today.
please, one question, please. One.
Okay.
One. Now, does it mean that for every cancer, for instance, cervical cancer, colon cancer, stomach cancer, skin cancer, there is a particular codon. There is a particular codon that
Yes.
is being screened in the human DNA. So if there is a known codon for every cancer, even those that are yet to be discovered.
We call
What if
it
it's what
markers.
if
We
a
call it
new,
markers.
yeah, markers? What if
Every
there is a
every
new,
disease has its own marker. So it can either be in the form of gene mutation, It can either be in the form of chromosomal abnormalities. So if you are looking for cancer, there is a particular marker they are looking for. A cervical cancer has its own marker. Breast cancer has its own marker. In one of our groups, we have a research groups as a result of our last session. We have this group are working. We are working on BRCA1 and BRCA2 genes. There's another group that is working on P53 genes. Yesterday's presenter is one of the leaders of the P53 gene group that they are working on that, working to do a review on the P53 genes. So there are different markers for different cancer types. So as a bioinformatician, this is when you'll be talking about that in transcriptomics or epigenetics, whereby you'll be studying the okay, we are still going to go to that when we talk about cytoscape, gene ontology, and gene enrichment analysis. So I am optimistic that we are going to benefit a lot from these sessions. So every cancer cell or every cancer has its own marker, and if you are a bioinformatician, what you are looking for those markers that distinguishes a particular disease. Even malaria has its own marker. So malaria, it has its own marker in your blood or in your system. So it's not just the a clinical manifestation of the disease that tells us that you have a disease. Like Angelina Jolie, that I cited in the exam, she never had that the pathological effects of the cancer she had. It was just as a result of screening that they discovered that she had that gene, that faulty gene. So she was able to do that through bioinformatics, and she was able to do that through bioinformatics screening. So they were able to notice that, Angelina, you have this faulty gene. Or if you do not remove it now, it is going to take away your life. So that is what bioinformatics does for you. And as a researcher, as a bioinformatician, you are not just you are also an epidemiologist. You are also studying the occurrence or the effect of a particular disease. So the potentials are so numerous. So, and I hope that I've been able to answer the question. So doctor, please, you have the floor. You can share the screen for the next presentation so that we can move on. I would answer the questions that has been posted on the comment sections one after the other. So please, uh, you can check the answers as you. If you have no answers, you can write questions. You can write it out. Then I'll try to answer them accordingly. So, doctor, please, you can share your screen. I think I've stopped sharing.
Okay. Right, can you see my screen now?
Yes.
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 Thank you. Okay. You're welcome again. So I'll be taking you through next generation sequencing. And to begin with, based on the various questions that have been asked, maybe I would like to do a kind of introduction to what we call the central dogma itself to help us to understand better why we go for sequencing. Okay, usually for every biological function in the body, there is most of the biological functions as a result of proteins, which are synthesized as enzymes. Enzymes are protein in nature, or some other, most chemical reactions in the body are regulated by these enzymes and which are protein in nature. So the body, as we all know, the DNA encodes all the body functions, all the biological functions. So this information that is in the DNA is in form of nitrogenous bases, like these nucleotides that we get from our sequencing. So usually how the body gets to regulate or control this biological function is through coding the information that is in the DNA in form of these sequences. These nucleotides are transcribed, the ones you have in your DNA, the codes, are transcribed into your mRNA, and this mRNA will now be further translated into protein, appropriate protein to carry out the function. So that's just the link between the biological functions we notice and the information that we have in our DNA. So going forward, so if there's any alteration or malfunction that is noticed in the body, so like for instance now, during most of our own analysis in biochemistry is majorly to detect biomarkers for a particular disease condition or some changes that is noted. So once this biomarker has been recognized during our lab analysis, so maybe we notice this protein, so it calls for querying the DNA, because we definitely know that this information that we are having, like noticing an abnormal concentration of a particular enzyme in your serum, so it calls for querying what something must be happening at the level of DNA, which codes the information that we desire, like this is the expected function. Like for every normal individual, there's a level of enzyme or analyte or a biomarker that we expect, like at a particular point. So during disease condition, that's why most times your patients or people who are having the condition undergo this. The first thing is to screen their blood. So when they see these changes, so they know there's definitely something wrong. So that calls for DNA extraction and subsequent sequencing of the DNA to see, oh, where is this coming from? So by the time that is done, then this is now going to be compared with a reference, a normal DNA, like this is what is expected of a normal individual. This is the nucleotide sequence that is seen in a normal individual that is not sick. So then they can compare and say, oh, this is where the changes are occurring and the like. So that is where sequencing, the idea behind sequencing DNA comes from. So now let's go to introduction to the next generation sequencing. So the first thing to going back in history is the prominent scientist Frederick Sanger. He was the first person to carry out DNA sequencing, and that was between 1918. So he lived between 1918 to 2013. So, and the first genome, like the genome is the total genetic makeup of an organism. So the first genome that was sequenced, the whole genome that was sequenced was the bacterial genome, bacteriophage genome. And this was done using the shotgun sequencing, which I would still highlight a bit more on as we go on. So other methods of sequencing were published by other scientists, which include Alan, Maxam, and Walter Liebert. So then Sanger also developed, he was, in addition to being the first person to sequence the DNA, he was also the first person to sequence the protein, and that was how he got his 2 Nobel Prizes. So the shotgun sequencing that I mentioned earlier on is just a laboratory technique for determining the DNA sequence of an organism's genome. The method involves randomly breaking the DNA after extraction is broken down, is digested into small DNA fragments. These are now sequenced individually. Like I mentioned, we want to see where the problem is coming from, depending on our objective of research. So a computer program now looks for overlap in the DNA sequences, using them to reassemble the fragments in correct order to reconstitute the genome and then map out for the differences and map out the differences. So this is just a pictorial representation of what sequencing involves. This is the genomic DNA. So after it is being shared, then it gives different fragments of DNA after digestion. Then you can see then these are the sequences. These are the various reads that we get from sequencing our DNA. And this is just an alignment, like assembly of the different reads that we have from this whole long DNA chain. So it has been broken down into pieces, and this is the assembly. So from first generation sequences to NGS, so usually, like I mentioned, the target DNA is fragmented and cloned in first generation sequencing anyway. So what happens in first generation sequencing is that the DNA is fragmented and cloned into plasmid vectors as part of the sequencing technique. So in this actually involves taking each individual fragment and now cloning them. You can imagine how laborious it will be and the time it will take to do that. So after that, the DNA is sequenced using the cyclic chain termination technique, and also it can be the radioisotopy can be used to detect some colored DNCPs. Then for the second generation, by time the sequencing technique is advancing, so by second generation, so what we have is a sequencing technique that involves the use of emulsion PCR, that's the polymerase chain reaction, and the bridge PCR are now employed in the second generation. Following these procedures, and there are procedures for it, which you can definitely check on later. So then for the third generation sequencing, it differs from the second generation because the sequencing techniques have now been created by numerous organizations, and these organizations are now using different, like higher throughput technique to make it better than the second generation, but it still lies on the basis is still on the second generation technique, but just a little advancement to make it better. And it gives a closer inspection of the target DNA. So this is just like a schematic representation of the schematic representation comparing the Sanger sequencing with the second generation sequencing. And as you can see here, there are a lot of differences for the Sanger sequencing. You can see that initially when the different reads are amplified using PCR, but here there is like an overlap, hybridization of fragments, like not taking the fragments one by one, and which makes the results better, and there is much output from the second generation sequencing. And so this is also a comparison between the first generation, second generation, and the third generation sequencing. We can go through it later once we have the slides. So the first generation sequencing is under it we have the Sanger sequencing, the Roche 454 sequencing, then the second generation sequencing, Illumina, the live ion torrent, then the third generation sequencing involves the Pacific Biosciences and the Oxford Nanopore. So these are this picture is just telling us like the advantage. So we'll still go into it in depth, but it's just like a snap into it. Like the first generation sequencing, the scientists involved are like hundreds of scientists come together to carry out the research. The machines are also in hundreds. It costs about $3 billion and it takes like 10 years. So going on to the second generation sequencing, to sequence DNA, you can two scientists can do that, and the machine is just one. Then the cost has reduced drastically from $3 billion to between a range of $5,000 to $10,000. You can see the huge difference, and it will only take 2 weeks. You can see the difference, 10 years from 10 years to 2 weeks. Then for the third generation sequences, there is still one or two scientists, the machines still remain one, And the cost, look at the cost now. We're now having $1,000 in hours. We can have our sequence. So, and these are different platforms that we mentioned earlier for the sequencing platforms. So, the Sanger versus the NGS, the new generation sequencing. So the sequencing by synthesis, Sanger is a sequencing by synthesis using labeled dideoxynucleotides or primers. Then the main reason for moving to NGS from Sanger is that there's a much higher throughput, the price is lower, as I explained earlier, and the speed is faster. So all are consequences of miniaturizing the processes. And what we mean by miniaturizing, like you see now, instead of having 96 wells per run in the Sanger technique, the number of sequences per read can be increased a lot. For the second generation, as you can see, I don't think this is the second. The iron torrents, it's from 1,000 to million of bits. Like you have more, it has more capacity because bits are used here. Then for the Illumina, you're having billions of colonies, thousands. Then for the PacBio, you're having millions of nanoparticles, which are what is being used there. Then also for the Oxford nanopore, there are not just hundreds of thousands of nanopore, but these nanopores can also be used, which will make it cost, like in terms of cost, it will be really reasonable to use the Oxford nanopore because you can still reuse the nanopores. Now, what are the pros and cons of the NGS? You can see these are the companies that we mentioned earlier. These are the different platforms that they use. So the pros and cons of those platforms are mentioned here, and majorly from the pros, we can see that it's the higher throughput, the accuracy, it allows peer-hand reading, the library prep, and you can see as compared with the cons, which is the cons means here, the speed here means that it's for Illumina, the speed is just the speed is not as fast as others, then the equipment price is quite on the high side. But for iTorrent, I think the price is more reasonable, and we can all go through this and see the differences in the different sequence techniques that we have. So comparatively, going by history and comparison with DNA sequencing technology. So improvement is mainly due to the miniaturizing that I mentioned, like then important difference between the short and long reads. Then also there's a big breakthrough with the use of Oxford nanopore. Like it doesn't require the use of DNA polymerase. Then what are the advice, the sequencing advice? The best higher throughput, if you are looking for the higher throughput, like good quality, and you go for the Illumina, the NovaSec, of the NovaSec platform. Then if you're going on a small scale, you go for Illumina, the MySec, which is representing the MiniSec. Then if you want to consider speed, you want your sequence fast, then you go for the iron torrents or nanopore. Then if you're considering a long read and modification, then you go for the PacBio. Then if you're considering long reads, you go for your nanopores generally. So that's that about NGN sequencing. Now let's go to mapping. This is just a pictorial representation of how so after your DNA has been fragmented into short reads, then you can see these are the short reads, then they are assembly. Then we now need to map it with the original genome. So the mapping is just a process of aligning the vast amount of reads that you have from your DNA fragmentation against a reference sequence. So that is mapping short reads to a reference genome to predict the locus where a read comes from. And like I mentioned, like we want to know what the problem is in the entire genome. So it's not all the reads that we need to map with the original one to be able to see the differences. So the result of the mapping is the list of the most probable regions with an associated probability. So this is an example. Let's say this is a read. I tried bringing it out in the form of something we can relate with, not sequence. So let's say the reference is good morning, beautiful world. And after this has been broken down, this is your DNA. So this was broken down, which is your which was broken down into small reads. So we have good, we have wood, we have different reads. But at the end of the day, all this came from the initial genome, which is the good morning, beautiful world. So like I said, these are the reads. So this is the reference. So when you are mapping, this is what mapping does. You try to align. So this is the reference at the bottom. You see, we are bringing the, so look at it. We are aligning the different reads like this. That's what mapping does. So what can we find with mapping? So you have your reference one in mapping, then you could see a notable change. Instead of having good morning, beautiful world, you can see here what we have is good evening. So this EVE is cause for attention. So we can begin to see, oh, maybe this is why we noticed whatever it is that made us to go for sequencing in the first instance. So this EVE is telling us definitely this is a suspect, like we could probe it further to see if this is what is responsible for the changes that we noticed initially. Then we also have SNP, that is single nucleotide polymorphism. Here, what you can see, instead of having good, so a single, so all these letters represent our nucleotide. So your G here has been replaced with N. So when you have just one of the nucleotides being replaced, that is what we call the single nucleotide polymorphism. So that's why we're having mood, morning, beautiful people, instead of having your good morning, beautiful people. So what we can also discover, detect with our mapping is, because we also detect insertion. In insertion, you're still going to have the good morning, beautiful people, but you're going to have extra non-mappable reads. Like you already have your complete read, but there's an extra that there's nowhere to map it. So that's what you call insertion. Then deletion, when there's something going away. You can see here deletion, what we have is good morning world. The beautiful has been deleted. So some of the nucleotide will be missed. So then you could notice that through your mapping. That would be what's the things that you noticed earlier. Then it will also have inversion. If you look at the inversion, here we are having beautiful coming from your, not arranged. The letters are complete, but in another order. Also the world, you can see. So that's what basically, like, in a way we can relate with. This is what happens in the mapping world. But you just have to, these letters will be replaced with our normal nucleotide sequence. So now, going on to the NGS5 format. So what are the next generation sequencing format? What are the formats that I recognize? The first one is the FASTA, like the FASTA format. So it's just your, sorry. So it's just a text file containing multiple DNA sequences, each with same text. Some part of the text might be just a name, like it's just your normal text. Like I mentioned, the sequences that you download from your data banks would usually be in form of your FASTA, and the A there is just standing for whole, meaning it could be your protein, nucleotide, or anything, your protein and nucleotides anyway. So these are, so it could be any of the sequences that you decide to download. So all come in form of the FASTA file. So the form in which they are downloaded, the file that contains them is the FASTA file. Then the FASTQ files are just like the FASTA file, but they also, in addition to the FASTA file, usually the FASTQ are a result of your sequence that has undergone some quality check, like trimming and things like that. So you like it has undergone some quality check. So you have some information telling you about the quality check that the FASTQ has, well, the sequences has gone through. And this, most of your sequencing data from your data from your sequencing machine or technique would always, most likely, because most of them have a quality check attached to them, the NGS files. So most of them have quality check attached to them. So they always, most likely, they come in form of the FASTQ. Then the SAM file, these ones are telling you that the files are, in addition to the sequences, they are having several sequences, but they are aligned already and scored against a template that is the reference. Then the BAM is just the binary version of your SAM file. Instead of having your nucleotide, seeing your nucleotide sequence, it's represented in form of a number. And it's just a combined binary format of your SAM. However, it can also be unaligned. So when it is unaligned, like you have your FASTA, which is just your normal sequences that are not aligned, then your FASTQ. So you have your SAM, which is the aligned sequences. So when you are now having a binary format of an unaligned sequence, it will not be this time around. It's not it's not the binary format of your SAM. It is the binary format of your FASTQ, because there the sequences are not aligned. Then you have your variance call format. This is just a file that has been aligned, whether your SAM or your BAM. Then in addition to the aligned sequences, it specifies a local where the widths disagree with the standard reference. So by the time you open your VCF, you'll be able to see, like, it's in addition to, by then you would have located the differences, like the probe in your sequence. So what does NGS quality control do? It checks the size, the number of reads, and usually it checks the residual presence of adapters. Sometimes in addition to your normal sequence, there are adapters. Some still contain adapters. So when you do quality control, you check for these adapters so that it can be trimmed away because it's not part of the original sequence. Then it's to check for the unexpected technical biases or unexpected biological biases. So that's basically what the quality control does in NGS. Thank you for listening.
Thank you so much for the presentation. That was pretty much loaded. So do we have any questions?
Yes, I do. I have a question.
Okay, go ahead, sir.
Okay, let me first by saying a big thank you to Dr. Suliat. She has done an excellent job. I've listened to different presentations on sequencing, new generation sequencing, but there is none that has made it so simple this way. Thank you so much. It's well appreciated.
She
My
deserves an
question
award.
is, she has an award.
I said she deserves an award.
I'm telling you, sir, the work was too sweet. It was too sweet. I had to be dealing with people that were disturbing me. Get away, get away. I want to hear this. Thank you so much. So I want to say something. As Africans, sometimes when you see things that are very fast, they make you afraid. Oxford nanopore. Are you sure? Sometimes when I sit down and I think about nanopore sequencing, I'm always afraid that the speed of this thing, am I sure that there won't be any deletion at all? Will it be as accurate as using Illumina? So please, I want you to help me with that. Help me with my bias. My bias is that the Oxford nanopore is too fast. Am I sure that if we make use of it, because now regularly now in Nigeria is becoming somewhat popular, we are having Oxford nanopore around now. So institutions are trying to buy, like my own institution, we intend buying our own before the end of probably the year. So I'm afraid that once we start using this thing, am I sure that the data from it is as accurate as using Sanga or using Illumina? Thank you so much.
Oh, I don't know. I've not used it before, so I'll find it hard to answer. All I can say is that that's why we have the pros and cons of individual techniques. So I think before venturing into it, you could also read more about it and be sure that it depends on what you want anyway. I've not used it, so my knowledge on that is limited. So let me just say that. I can't really say much on it.
Okay, ma, thank you so much. But I will still be disturbing you. Thank you so much. I really appreciate it.
You have to pay for that private consultation then.
Don't worry, sir. Don't worry, sir.
I'll be the accountant.
Yeah,
I'll be
no
the
problem.
accountant.
I will see you before seeing her. Don't worry, sir.
Good.
Thank you so much, sir. Bye, sir.
Any other question? Okay, I think if there are no questions, we can have like 10 minutes break so that we can come back for the last session for today and then we can call it a day. So before we do that, I would want to make an announcement. So tomorrow's session is going to be about the Ensemble browser genome. So one of the topics or one of the um, themes to discuss tomorrow will be a recap of the last session we had. So it was an 8-hour long session. So I would also provide the link to the YouTube video for that. So the Ensemble browser genome is also used extensively for population studies, biological studies, epidemiological studies, and the likes. So it's a genome browser, and you can use it for a variety of purposes. So, so that for those who are just new to the browser, we would provide a link so that we can go through them before tomorrow's class. So the links to tomorrow's class too is going to be on Zoom. So the links, inshallah, God willing, will be provided so that we can work with that too. So we'll be going for a 15-minute break, and then we'll come back to have the conclusion of the session. We'll see you in 12 o'clock Nigerian time. So thank you so much.
Thank you so much, sir.
So we're about to begin the last session for today. So we are awaiting others to join. We're around 20, about 30 presently. So so you're welcome to the third section of today's section. Uh, and we'll be talking about the introduction to epigenetics. So, um, how would I handle that? Like epigenetics is also a very, very important aspect of bioinformatics. That it's an advanced stage, or should I put it advanced stage? In our last, the third cohort, we also made a little bit of introduction about what epigenetics is and its importance to biology, bioinformatics. So need I mention that relating to bioinformatics, there are different aspects that you might choose to venture into. For example, I am particularly focused on the genomics part of bioinformatics. My other colleagues are they are focused majorly on the other part, which is transcriptomics and epigenetics. We also have another colleague of mine who is into metagenomics, which is a branch of genomics, so that I am focusing on. And there are also others who are into population studies. Presently, in my laboratory, I am the only one working on metagenomics of mosquito and the symbionts and bacteria. And they focus majorly on population genetic studies. So they don't have anything to do with all this RNA sequence or mRNA sequence and the like. So it's basically DNA sequences, full genome, CO1, ND2, and the like, ITS2, microsatellite. So that's what we deal with majorly in the lab. So I'm the only one focusing on a certain level. I'm not going deeper into population genetics, although my master's program, I also did on population genetics, and I also still do population genetic studies. But what I'm trying to pass along is that there are different aspects of bioinformatics that you might want to choose and delve deeper into, depending on your choice, depending on your wish, or depending on your interest and area of research. So that's why today's class is also multifaceted. So we talked about a little bit of genomics, and now we are diving deep into the area of epigenetics. So the floor is open to resourceful resource person, doctor. So you have the floor, ma'am. Thank you so much.
Thank you, everyone. Welcome back. So now I'll be speaking on epigenetics, and I'll just be disclaimer. I'll just be introducing us to the concept of epigenetics, so that we have an idea of what epigenetics means when we hear epigenetics. So let's just continue from where we stopped the other time. So we basically go for sequencing to probe changes in DNA. So let's now assume after probing, like sequencing our DNA, and there's no change, or there's no notable change in our DNA, and yet we're noticing some, there were some phenotypic changes that we saw that made us query the DNA. So that's where epigenetics comes in. So literally, epigenetics means above or on top, like epi, above, on top of genetics, like beyond DNA. So, and this means information coded beyond the DNA sequence, such as in covalent modification due to modification that occurs to the DNA or the chromatin structure, like the chromosome. So practically, epigenetics just describes the phenomena in which genetically identical cells or organisms express genomes differently. Like they have the same genome, but their expression of the genome when it comes to their phenotype is different. So epigenetics helps to study heritable traits that cannot be explained by changes in DNA sequence, but due to changes in chromosome that ultimately affect gene expression. Like now, what happens basically, okay, let's just move on. Let's see. So this is just a typical example of an epigenetic phenomenon. These are twins with the same genome, and we can see that they don't look like they are actually identical. So expecting them, like with them having the same genome, what would expect on a good day is that they will be looking alike. So this is also another. These are cats. These two cats, they have the same genetic combination, but looking at them, we see that they are different. Their phenotypic expression is different. So this is the mother and the baby cats with the same genome or different phenotypes. So basically, from history, so many people have said that the diet of a pregnant woman could result into an epigenetic outcome. Like even with the fact that it's expected that the genome of the child will be identical to that of the mother, but the diet of the mother during pregnancy could result into the modifications that makes the child to express different phenotype from the mother. So some also claim that the father also counts in this. The diet of the father could also count, and a story was given of a generation in which there was shortage of food for the grandfather, and this shortage of food, for the effect of this shortage of food extended to the lifespan of the grandchildren. How? because it was noticed that the grandchildren they had longer lifespan due to the fact that the shortage of food that the grandfather has endured during his own lifetime has brought about a kind of modification to the gene of the grandfather, which not the nucleotide sequence this time around. Because eventually what happens is that in epigenetics, our gene, our DNA is when the DNA is unwound, like it's not in its compact region, like in the nucleus. Considering the length of the DNA, if you compare with the size of our nucleus, this nucleus won't be able to contain the DNA. So this DNA has to be compacted in form of what we see as a chromosome. So because if the DNA is stretched out, it's several meters long. So in a way to compact the DNA, the histones, some other compounds like histones are used to carry out this compacting of the DNA. So, and these histones are also protein. So during this compacting, definitely this histone has some branches and has some side chain branches like structures like hydrogen bonding and things like that could interact with the structures of the DNA. So bringing about a kind of modification to the gene. So in the case of this grandfather who has, and usually histone can also undergo what we call post-translational modification. You know, this histone is also a protein that was synthesized in the body. So meaning that after the protein has been produced, it can still be modified due to some changes in the organism or individual. So when there's a change in this modification, like in this histone, due to the condition the person is predisposed to, this could result, that change in histone would also, it would eventually affect the interaction of the histone with the DNA, because it is the histone that rounds around the DNA. So this kind of interaction could bring about a notable change, and that could be the explanation given to some notable changes, even though there are no specific change in DNA sequence, but there are some adaptation technique that the earlier generation have used, could be passed on to the subsequent generation. So the epigenetic change is a regular and natural occurrence, but can also be influenced by several factors, including age, environment, lifestyle, like I mentioned, disease states, and epigenetic modification resulted from external and environmental factors, still repeating the same thing. So not all genes are active at all times. Yes, one thing epigenetics does is, due to the winding of this DNA, like compacting it into a small structure that can be contained in the nucleus, it's, you know, for DNA, I mentioned earlier that for the information in DNA to be coded, it has to be transcribed into mRNA. So sometimes the coming together, like condensation of this DNA, compacting it, makes it less available for transcription. So some transcription processes will not occur fully, and that could lead to a notable change, because eventually we're going to be having a protein different from the one that we should have, because once the DNA is not opening up due to compaction, the compacting effect of the histone that has helped to compact it, so it doesn't open up for transcription to be done efficiently. So definitely we'll know what we have in our mRNA will be different. Like, it's not the expected. You know, we could predict this is what we have using the complementary base pairing. So by the time we compare the expectation, you see, so, and you know, the information in the DNA is coded there. So probably when it gets to a particular nucleotide, the mRNA could not transcribe, the DNA could not be transcribed into mRNA. So definitely this will affect, because it is the mRNA that would eventually be translated into our protein. So, and it is the protein that will bring about the phenotypes that we see. So this is one of the way where epigenetics come in, and majorly these are due to modifications as a result of the methylation of DNA, which occurs indirectly due to the modification of the histones. So generally the chromatin, they are of two types. They have the heterochromatin, like the way the DNA is being packed. You know, we're talking about the chromatin. And when we're talking about the chromatin, we're still talking about the DNA. It's just the packed DNA now. So we have what we call the heterochromatin, and we have the euchromatin. So for the heterochromatin, it is tightly packed. So then it is a gene-rich region because it is easily accessed for transcription. It is a gene-rich region, but it is not accessible as compared to the euchromatin, which is an open chromatin, which can easily be accessed, which can be transcribed easily compared to the heterochromatin. It is only the heterochromatin is packed and is not as open as the euchromatin. So basically, this is how epigenetics. This is just the mechanism of epigenetics. This is how it affects the gene expression. There could be it could affect it through histone modifications. That's the protein I mentioned earlier that helps to compact the DNA structure. Then DNA methylation. This DNA methylation is still somehow linked to the histone modification. So we'll see as. So this is just an explanation that I gave earlier to why we are having your DNA packed into interacting with histone. Like usually DNA are always compacted via interaction with proteins, which are the histone proteins, because you can see the nucleus is just 10 micrometer in size, and the human DNA is 2 meter. So how can we now put this 2 meter DNA inside the human nucleus, which is just 10 micrometer? So this has to be done by compacting the DNA into a size that can contain the nucleus. And this is done with the interaction with the histone protein. So I've already mentioned the types of chromatin. The euchromatin is transcriptionally active and it is less compacted. These ones are less transcriptionally active, and these are the types that usually give us the epigenetic changes that we notice. So, and it has been, all levels of condensation of chromatin have been indicated in controlling the accessibility of the genomic DNA, whether highly compacted or less compacted. So I think basically I've explained how we have it. So let's just go deep a bit, not so deep into the chemistry of the histone and how it results into the changes that we notice in our phenotype. But I couldn't prepare the proper slide for this. I had to just use this material that I have. Okay, so we want to talk about the chromatin structure, like the resulting structure we have after our DNA has been compacted. So there is the primary structure is just the linear arrangement of features such as the nucleosomes on DNA, like how the DNA is arranged. The nucleosome is just your DNA and your histone. So how these are arranged, that's the primary structure. It's just like when you have the primary structure of your protein, meaning that the the just the straight sequence of your amino acid. Then the secondary structure is by interaction of the nucleosomes. So now the first structure is just alignment of the DNA with your histone in straight, like just a linear arrangement. So by the time you are having your secondary structure, this linear arrangement has been folded. So there is a level of interaction that will be expected at the secondary level of interaction. Then the tertiary structure is just an interaction between the secondary structures. So, and we can see here, these are just, this is the primary structure, the secondary, and this is the tertiary structure of the chromatin. So this is just the primary structure showing us the nucleosomes. And nucleosome is just, like I mentioned, is the DNA plus the histone. So this is the core, the histone acting as, these are linkers. These are the core DNA structure. So like I mentioned, the nucleosome is histone plus DNA. Nucleosome is a basic structural unit of your DNA packaged in, as it is packaged in eukaryotes. So consisting of a segment of DNA wound in sequence around 8 histone proteins. As you can see here, these are the histone proteins, and this is our DNA. So the nucleosomes are obstacle to transcription. Like I mentioned earlier, this modification, like compacting the DNA, affects the transcription of the DNA. And so let's go to the histones. The histones are primarily, they are chief protein component of the chromatin. Then histones are protein around which DNA can be wound for compaction, like reducing its size. Then for gene regulation, histones have been implicated in gene regulation too, because they can actually undergo post-translational changes. So gene regulation can be targeted at histones. Once there's a modification done to histone which interacts with our DNA, we can thereby regulate some genetic expression through the modification of our histones. So histones are highly alkaline proteins found in the eukaryotic cell nuclei that package, and the eukaryotic cell package like we have it, and it helps to package the DNA into structural units called nucleosomes, and they are generally of 5 families, the H1, H2, and the likes are seen. So now, how do we now move from genomic organization, because that's the expectation, that's what we think histone is actually doing for us, to genomic regulation? Now, so histone modification. Histone undergo post-translational modifications that alter their interaction with the DNA and the nuclear proteins. So that's basically how your histone gets to affect the transcription and eventually the translation processes, because it can actually undergo post-translational modification. And this modification includes methylation, acetylation, phosphorylation, and the likes. But the common one that we are most used to hearing, even if we are not so sure of the function, is the methylation of the DNA. So histone modification happens in diverse biological processes such as gene regulation, DNA repair, and the likes. So there are different classes of the histone, the conserved and the highly conserved, and the ones that are not conserved. The linker histones are the ones that are not conserved, but the core histones that are involved in the compaction of the DNA are the conserved ones. So basically, that's the genesis of epigenetics. Let me just stop here and I wouldn't bore you with the deep chemistry of epigenetics, since this is just introduction. Thank you for listening. Hello?
Thank you so much. Yeah, hello. Thank you
All
so
right.
much for the wonderful presentation. It's a Um, it's indeed very fully packed. So from what we've listened to so far, so we've been made to go through the several aspects of the importance of epigenetics and the potential it holds for bioinformatics. So one of the ways in which epigenetics holds importance is the activation of genes. Just like she mentioned regarding during the unfolding and the transcription to proteins. So there are some genes that would not be activated. So there are still science is still trying to unravel the reason why some genes are activated and some genes are not activated. For example, just to simply put it, uh, the um, the genome will be shared. Sorry, the slides will be shared. For example, when we compare African populations and Black populations in America, we noticed some there are remarkable differences in the genome composition. So, and this is attributed to epigenetic differences because ordinarily those that are blacks in America, they have ancestry traced back to African origin. But because of the environmental changes and the like, so these so there are some genes that are switched off and there are some genes that are switched on. So in the individuals regarding despite regarding the geographical location that everyone that they have. So also is food. So also is the age has impact on that. So I'm very sure that we will not be able, our presenter will not be even able to exhaust the full scope of what epigenetics is and how bioinformatics comes into place within the short period that we've given her. And because it's kind of a more advanced level of molecular biology, so it will be difficult for a lot of people to grasp the full extent and the scope of what it is all about. So, but notwithstanding, I would want to leave the floor open for questions. So if we have any questions for our lecturer regarding the last presentation, then please go ahead. Any questions?
Yes, I have a question.
Thank you.
So I want the presenter to throw more light on DNA methylation.
Doctor, you have the floor, ma'am.
Okay, sir. Sorry, what's the question?
You, you, you have to shed, please help us shed more light on DNA methylation, ma'am.
Okay, DNA methylation is just from the word methylation, the process by which DNA methyl group, methyl, is added to a DNA molecule. That's just what DNA methylation means.
Do we get
I
that?
want, yes, we do. So I want to ask if methyl is added to
I want
DNA
to have
methyl,
I have a
what
question.
does it cause? What's the effect? Does it have any phenotypic expression? And is it advantageous or is it disadvantageous to us?
So basically, to add to what Doctor has explained, so DNA methylation, when the methyl group is added to the DNA, so what they basically do is that they change the activity of the DNA segment without changing the sequence. So we all know that the DNA molecule is highly like they are wound around each other to form the chromosome as we have them. So, but if the methylation goes wrong, it would have an effect on the DNA itself. So, for example, when located in a gene promoter, so the DNA methylation typically acts to repress gene transcription. So it doesn't allow the gene or the DNA to go into transcription process. So which also signals either DNA activation or inactivation. So if it is activated, it depends on the type of gene you are talking about or what you are trying to work with. So if it is activated, it might be a benefit for you. For example, when we do molecular analysis whereby you do each shock protein, each shock, you see an upregulation or downregulation of a particular gene. So upregulation or downregulation of a particular gene. So this is as a result or as an effect of similar processes like DNA methylation. So when a gene is upregulated, so it might be as a result or as a reaction to a particular intruder into the system of the organism. So and if a gene is downregulated, it might be as a reaction to something not happening. So DNA methylation is also impacted in a lot of biological processes, and such is it is included in the process, key process like carcinogenesis and aging process. So and S chromosome inactivation and the like. So it's very wide. The impact is wide. So,
Okay, my
doctor,
question. I
if
have
you
a
have
question, please.
any addition, if you have any addition to the answer or
No
removal.
addition, sir. Sorry.
Oh,
Can I
okay.
go ahead? I have my question, please.
Yes, yes, yes. Please go ahead.
Okay, my question is, how do you study epigenic, how does this epigenicity, how do you assay it in the lab? That's what I was trying to ask. How do you assay it in the lab? What are the studies you study so that you can, just like we have other bioinformatic procedures you can study in the lab, how do you study epigenicity in the lab?
Okay.
So it can be. Oh, sorry. Go ahead, ma. Doctor.
Please go ahead, sir. I'll talk after you. Don't worry.
Okay, so you can. There are different ways in which you can detect DNA methylation. So, and one of such ways is mass spectrometry. So, it
Okay.
is one of the reliable analytical methods to detect DNA methylation. So, when the first DNA structure was discovered by what's her name? For, I've forgotten the name of this researcher, but it was Francis and Crick that won the Nobel Prize for the structure of DNA. So the DNA was discovered through this mass spectrometry. So and one of the ways in which DNA methylation can be denoted is mass spectrometry. In fact, it's the most reliable analytical method to detect mass spectrometry. So there is another method that is called the chip on chip assay. So that one is you are going far, 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 far. There is also another method of pyrosequencing. So pyrosequencing is implicated in one of the methods of next generation, say I think second or third generation sequencing method, or even the first generation. So is implicated in that uh, process too. So there are many many ways in which you can detect DNA methylation, but I mentioned the most popular one. You can also do it through methylation specific PCR. So we all know what PCR is. So, but this one is customized for methylation purposes. So that's how you detect DNA methylation in the lab. So and you can do any other analysis.
Okay, let me also ask, at the moment now, are there labs in the world that are doing it? Are
Of
there
course.
places?
Of course, there are many labs in the around. I don't know about Nigeria presently actually, but there are many labs that are doing methylation process. Any lab that has any chemistry lab, majorly chemistry or physics lab that does mass spectrometry, they will definitely have what it takes to do to detect DNA methylation or any other abnormality in the DNA. So I think that answers that. Hope Doctor, if you have any addition,
Okay, nothing much. Also, they use DNA enzyme digest to also detect DNA methylation, in addition to what he has said. So this method is basically used, is based on DNA endonucleases. So these DNA endonucleases do not cut methylated DNA. So digestion of specific DNA target sequences by this enzyme generates DNA fragments of different lengths, which may be sequenced to determine the extent of methylation using other techniques such as your ELISA and the likes.
Sir, I also like to add to it another way of determining DNA methylation, sir. Can I go ahead?
okay, please go ahead. Please go ahead. Yes, please go ahead.
Yeah, we can also determine DNA methylation through bisulfate sequencing or metisync. This is a way of determining where on the
Please
genome
come
of
again.
DNA. I said through bisulfate sequencing
Okay,
or
okay.
metisync. This is a way of determining where on the genome the DNA has been methylated. Methylation is an important epigenetic modification that also affects which proteins are being expressed in the cells. And these methylation marks or methyl groups can be passed on from one cell cycle to another as cells divide. How can it be achieved? The DNA is being split into two identical samples. Then it will take two, then you take two very small samples or aliquots of DNA, and then you treat one of them in a special way called bisulfite conversion. This biochemical process converts all of the Cs that are not methylated to use, that is the cytosine that are not methylated to uracils. It is worthy of note that methyl groups are always attached to the cytosine. So the methyl groups are usually attached to the cytosine. That is the only nucleotide they get attached to. So this process now gives us two identical samples where we have converted in one sample the DNA in a special way so that many of the Cs are now used. And then we sequence again and we have to compare those, and those one require very specialized programs that do not just comparison but also the alignment, because that converted DNA now doesn't really match the genome very well. So the reference genome are the one that are usually aligned to. So it's just a way of just knowing that, okay, at this part there is a methylation and at this part there is no methylation. So by the time all of those things are done, then comparison is actually made. But I discovered that some of these things we get to understand also even as we read more about. Thank you very much.
Thank you so much. Thank you so much. Uh, I want to ask, is it possible in Nigeria
Okay,
for
this
those who have
process now, as long as we're able to get the procedures, then I don't think it should be difficult for us to do in Nigeria.
Let me ask him, does he have any lab in Nigeria that can do it at the moment?
I don't know of any. I also learned it somewhere, when I was learning
Because that's
DNA.
the reason why I asked. Because I have not any lab in Nigeria that I know there are places that have facilities that have mass spectrometer, but I don't think they have the procedures
And
to go about it.
once you can get the reagent, you just simulate the
I,
procedure wherever you are. I think if it doesn't really require any big instrument, it's possible, it should be possible.
and then I think in Nigeria we do things that will actually help, that will actually going to benefit from. If your problem is not related to DNA methylation, you may not have to think about determining it. It's when your baby research is towards that line, then you look for labs to see whether it can be done here in Nigeria or
That's
not.
true.
okay, thank you so much. Thank you. I've been corrected regarding the Rosalind Franklin. So it's X-ray crystallography, not mass spectrometry. So thank you so much for that correction. So so mass spectrometry for DNA methylation detection and crystallography for mass X-ray sorry X-ray crystallography for used by Rosalind Franklin for the structure. So thank you so much for the contributions. Any other contribution or question? I think no other.
I want to ask a
Okay.
question, please. It just
Okay,
came to
so
my
go
mind
ahead.
now. Yeah, my question is, I want to ask that the application of epigenetics, you know,
Hello.
is it? It looks as if maybe does it have more influence in plant breeding and genetics than in animal genetics? Because that's when we see a lot of aberration that may not necessarily be as a result of the genome. Because I discovered that when I was in school and then I was doing some genetics courses on crops, it looks as if they have so much aberration, tetra plus all of these things than the animals. So I want to ask that is epigenetics more related to plant genetics than animal genetics?
I
Doctor
don't
is a
basically.
question. No.
Okay, I don't think there is any specificity for plants in epigenetics. It's just that I think it was first noticed in plants, and plants have been studied, epigenetics has been studied more in plants. That's just the reason. I don't think there's any reason why plants are more it's not just peculiar to plants anyway. That's the answer. It's just that I'm not sure, but I think it was first observed in plants, and several studies have been carried on, several epigenetic studies have been carried out on plants.
Thank you so
Thank
much.
you.
I think in addition to that, it depends on what you're working on. You think everything revolves around that particular stuff. So I had that experience when I was working with mitochondrial DNA. When I started working on, I thought it's only mitochondria. So until I discovered that there is also cytoplasm, and the like. Okay. So it looks as though that it is more of present in plants than in animals. So, but I think the reason for that is that a lot of experiments can be done on plants easily than it can be done on animals. Regarding, for example, even humans, let me put it that way, in human, because you can have as much as you can kill, uh, you can like test run on different plants as much as you like, and that cannot be possible with human samples. So and even it is much more lesser in animal samples too. So I think that's the reason why it might look that way. So thank you so much for the question and the contribution. So without further ado, in order to save us time and save our data, so thank you so much for being here. The recording of today's session will also be shared to us. Yesterday's session, I've already compressed. I've already made the video. I will also share that too on our Telegram page and later on the YouTube page. Then tomorrow's details for tomorrow's class will also be sent tonight, God willing. So I will share that as well. So thank you so much for being a part. So if you have any questions, please don't forget that there will be a certificate after this course session call. So we would love every one of us to have it as a testament that we were here. So thank you so much.
Thank you
Sorry,
too.
please,
We
Uh,
appreciate you.
before you go, please.
Mr. Mark, then after
Yeah,
Mr. Mark, Mr. Elu, sorry,
it,
this
it, it,
Elu
Remy,
I
Remy.
wanted
Dodun.
to ask a question.
Okay,
Okay,
maybe
sir.
I should give
Hello,
them space.
After Mr. Mark, sir.
please, my question. Okay, my question is centered on that area of having a conserved vision of these areas that are not conserved. I want to be clear. When she was presenting, she mentioned the
We
stones,
can
the
hardly
ones
hear
that
you,
are
sir.
conserved, and the ones. Okay, hello, can you
Yes,
hear
yes,
me?
it's better.
Okay, when the presenter was talking about histones and the chromatin, she mentioned something about the area, the ones that are conserved and the ones that are not conserved. I want to be clear about that conservation um, manners of to some and to some that are not that just want to be clear. Hello,
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 sir.
hello. Did you get my question?
Um,
Or did you get my
Doctor,
question?
Yes, sir. Okay, you're asking, your question is if I get you right, conserved stones and the ones that are not conserved. So the core
Yes.
stones are the conserved ones, whereas there are some stones that are just linking one structure. You know, when we talked about the structures of the chromatin, we talked about the primary, secondary. So for the tertiary, we said linking of different secondary stones together. So there are some stones that they are just present as linkers. So these are the ones that are not conserved. Meanwhile, the real stone that is wound around the DNA, that DNA is wound around, they are conserved. Meaning, the only thing I can say about that is that the conserved ones means that they are not susceptible to modification. So when we want to really study the modification changes that we notice in our stones, we'll most likely go for the unconserved ones.
So does it now mean that the conserved ones are not studied? How do we now know the theoretical role these conserved ones will play
I can't
when
hear
they are
you
not being
clearly,
studied?
sir.
So I'm saying that does it now mean that the conserved stones are not well studied at this time?
They've been studied. If they were not studied, they would not discover that they are not that they are conserved. It is studies that actually shed light into the fact that they discovered that there was no changes in them. That's just meaning of conservation. So, but the ones that are not conserved, maybe they notice the modifications in them. So that's why the terminology conserved and not conserved. Right. I think that's the.
thank you so much. Mr. Edu, I'm very sorry, the
Okay,
name.
okay,
Uh,
okay.
Okay.
It's Elu Dodun, Remy,
El,
sorry.
El, okay,
Elu
okay,
Dodun,
Remy. Okay,
yeah,
Elu Dodun.
yeah. Okay, thank you very much. I really appreciate it. I'm really learning from this webinar. Thank you very much. Yes, but I have a little challenge because my system is not available, but I've been trying to follow the class because I know theories will later be a background for practical knowledge. But I got somebody's system and I wanted to try out the bioedit and then the mega yesterday. So I got a sequence. So I got a sequence and that has a reference genome and another genome, though it's confidential, you know. And I tried to I copied and pasted those sequences in Notepad, so I put them together. But the arrangement was not the best I wanted, but I discovered that there was no way I could make them to be on a straight line using enter key to arrange them because it was so plenty. So then after that, I was able to successfully upload it into the bio edit, but the stuff keeps processing and processing and reading. I was like, yeah, is it going to take hours before it's going to be able to process it? So I don't know what happened with that. Then I also tried Mega. I was trying to upload. I was trying to open the data on the Mega. It was only bringing the interface like a sub interface of detect edit. How can I now? Text editor kind of. So there was no way I could run the analysis of aligning it on the Mega. I don't know whether you get my question. So I was like, okay, what happened? Why is it that it's only bringing this surface of editor, the editor, and then I'm not able to use the align option
But
on the Mega. So those are the two things I encountered over the night.
so, how many sequences are your sequence?
570 something.
So you have to wait for BioEdit to finish running the alignment.
Like how many minutes?
You have to wait for hours.
Wow. I was so
It de-
confused. I was like,
it de- it depends on the strength. Even it depends on the strength of your system and your internet. No, you don't need internet. It depends on the strength of your system. So if you are running 500 and something sequences, and each sequence is about 1,000 base pair long, so you can just, it's like, for those who are in engineering, it's like you are running a simulation process. So you have to wait it out. So it's not a problem if it is still running. Even till now, if I run alignment on BioEdit, about 15,000 base pair long sequences, and I have like 10 or 20 of them, I know I just have to wait for it to finish running before I see my result. Then secondly, regarding the Mega, so Mega will not upload your sequences except except it is aligned, except it is saved as a FASTA file. And the easy way to do that is very simple. Go back to your file. So after your txt file, you will see the name. Let's say you name your file NOBLEKINMAT. You will see dots. What is after it is txt. So if you want to change it to FASTA, you can simply change the txt. You rename the file. You rename it as dot the name dot FASTA FASTA. So through that, by Mega will, at least if your sequences does not have a problem, so it will upload it to the system. So then another word of caution is that probably you did not save your sequences right. On the first line of every sequence, you have to put a greater than sign, followed by the name, then under it you will see you put the sequence. So that's how you do it. If you do not do it that way, even BioEdit will not be able to recognize your sequences you upload. So that greater than sign is very, very important for you to put. So you can try those options, and hopefully you'll get a better result.
Oh, thank you, sir. But before you leave, I came a little late yesterday, when you were walking us through the use of BioEdit, and then you were actually using the sample sequence. So, but I could not get how you got it. So, and then I watched out for the recordings. It's not available yet because of the last size. So I don't know if you
No,
can.
it was the first day. The recording has already been sent.
Okay, yeah, okay. It was first day before yesterday. Yes.
Yeah, yeah. The recording has already been sent. You can check it out. Mhm. I think you can. That would be nice. Let me check. So we downloaded it from NCBI, and the process are very simple. So thank you so much. Do we have any contribution before we call it a day? I think no contributions or questions. So see you tomorrow. Thank you so much. This is the official ending of day 3 of NOBLEKINMAT for cohort by informatics training for beginner, intermediate, and experts. So we thank you so much for being a part from the beginning, and we hope that you stick with us till the end of the session. So thank you so much, and we would love to see you tomorrow. See you tomorrow. Bye bye.
Thank you very much. Bye.
You're welcome.
Thank you.
I thank you so much. God bless you.
Amen. And you too.